Number 10. Pandoravirus yedoma, the 48,500-year-old nightmare that shouldn't still be infectious. In February 2018, French microbiologist Jean-Michel Claverie pulled something impossible from frozen Siberian mud. A virus. Not just any virus. The oldest living virus ever recovered from permafrost, buried 52 feet below an underground lake in the Russian Far East. When his team revived it in the lab and exposed it to amoeba cells, the virus did what viruses do. It infected, it replicated, it killed its hosts, as if 48,500 years of being frozen solid was just a brief nap. The scale of what's happening in the Arctic right now doesn't fit comfortably in the human brain. 4 sextillion microbes are released annually from thawing permafrost. That's a 4 with 21 zeros after it. Every single year. The Arctic is warming 4 times faster than the rest of the planet, and 1/4 of the northern hemisphere sits on permanently frozen ground that's becoming decidedly less permanent. We're opening a biological time capsule we didn't know existed, and we have no idea what else is in there. Pandoravirus yedoma is enormous by viral standards. 1 micrometer long, visible under a standard microscope, shaped like a massive egg. It contains approximately 2,500 genes. Modern viruses that infect humans typically carry 10 to 20 genes maximum. This ancient thing is genetically complex in ways that make contemporary viruses look primitive, which raises an uncomfortable question. If something this sophisticated could survive nearly 50 millennia in frozen darkness and wake up ready to function, what else is down there? The permafrost isn't just cold storage. It's an oxygen-free environment that light never penetrates. Perfect preservation conditions. Viruses don't decay. They don't decompose. They just wait. And the world's permafrost contains an estimated 1,500 gigatons of carbon, roughly twice what's currently in the atmosphere. As it thaws, that carbon breaks down and releases as carbon dioxide and methane, which warms the planet further, melting more permafrost. A feedback loop that accelerates with each passing year. Number 9. Pithovirus Sibericum, the cork-shaped giant that redefined what frozen ground could preserve. When scientists first examined this virus under a microscope in 2014, they thought they were looking at a bacterium. It's 1.5 micrometers long, shaped like a thick-walled oval with a honeycomb grid structure and what looks exactly like a cork stopper at one end. The virus was extracted from 30,000-year-old Siberian permafrost, buried 100 feet below the surface in late Pleistocene sediment. Here's how they found it. Riverbank samples collected in 2000 were stored, then later exposed to amoebas in a laboratory setting. The amoebas started dying. When researchers examined the dead cells, they discovered them packed with giant virus particles. This became the standard protocol. Expose ancient permafrost to amoebas. See what kills them. The method has been disturbingly successful. Pithovirus Sibericum combines the massive size of giant viruses with a surprisingly compact genome. Just 600 kilobases of DNA, smaller than you'd expect for something so physically large. Its gene content resembles iridoviruses and Marseille virus more than other giant viruses, but it replicates entirely in the host cell's cytoplasm like megaviridae. It doesn't fit cleanly into existing viral categories, which suggests we've been categorizing based on an incomplete picture. What makes this discovery particularly unsettling is the implication. If a 30,000-year-old virus can remain viable in frozen sediment, then the entire archive of prehistoric viral evolution is potentially still active. Every pandemic that swept through Ice Age populations, every pathogen that infected extinct megafauna, every viral lineage that disappeared from the surface world might still exist in perfect stasis beneath our feet. Number 8. Mollivirus Sibericum. The soft giant that proved viral diversity in permafrost goes deeper than anyone expected. This virus came from the exact same permafrost sample as pithovirus. Same location, same age. Completely different structure, approximately spherical, 0.6 micrometers in diameter, with a genome encoding 523 proteins. 64% of those proteins are orphans, meaning they have no known relatives in any other organism. 16% show closest similarity to Pandora viruses. 10% appear to have been acquired from Acanthamoeba castellani through horizontal gene transfer. The fact that two entirely distinct giant virus types existed in the same sample shattered assumptions about viral preservation. Researchers had suspected permafrost might preserve the occasional hardy specimen. Instead, they found a rich archive of diverse viral families, each with different replication strategies, different structures, different genetic compositions. But Mollivirus sibericum has another disturbing characteristic. Unlike pythovirus, which only needs the host cell's cytoplasm to reproduce, Mollivirus requires access to the cell nucleus. It's as host-dependent as common viruses, including human pathogens like adenovirus, papillomavirus, and herpes virus. This similarity in replication strategy means Mollivirus represents a viral family type that could theoretically include human infectious members. The metagenomic analysis revealed extremely low concentrations of Mollivirus in the sample, just a few parts per million, which sounds reassuring until you remember that in the presence of a susceptible host, a few infectious particles are sufficient to trigger an outbreak. And climate change isn't just thawing permafrost. It's making Arctic regions increasingly accessible for mining, oil extraction, and industrial development. More human activity in regions where ancient viruses are being released in concentrations we still don't fully understand. Number 7. The mammoth wool viruses, proof that prehistoric animals carried pathogens we've never encountered. 3 viruses were pulled from a single clump of 27,000-year-old petrified mammoth wool found on the banks of the Yana River in the Russian Far East. Megavirus mammoth, pythovirus mammoth, and Pandoravirus mammoth. 3 distinct viral families, all preserved in the hair of an extinct Ice Age elephant. 2 more viruses, Pacmanvirus lupus and Pandoravirus lupus, came from the frozen stomach contents of an ancient Siberian wolf. These discoveries prove something researchers had suspected but couldn't confirm. Viruses survive not just in permafrost soil, but inside the preserved remains of dead animals. And as Arctic temperatures rise, remarkably well-preserved animal carcasses are appearing across the tundra. Woolly mammoths, steppe bison, cave lions, ancient horses, all emerging from ice that's held them for tens of thousands of years, all potentially carrying viral cargo. Human immune systems have never seen these pathogens. We've evolved alongside modern viruses. Our immune systems know their patterns, recognize their structures, carry genetic memories of similar threats faced by our ancestors. But a virus that infected a mammoth or a cave bear? We have no blueprint for that. No inherited defense. Our bodies would be encountering something genuinely alien for the first time. Number 6. The 2016 Siberian anthrax outbreak, when theory became reality and a child died from a disease released by melting ice. Summer 2016, Russia's Yamal Peninsula. Temperatures reached 35 degrees Celsius in the Arctic Circle, unheard of for that region. The permafrost, which had remained frozen for millennia, began to thaw rapidly. Beneath the surface lay a reindeer burial ground, animals that had died more than 70 years earlier from anthrax. As the ground thawed, Bacillus anthracis spores, dormant for decades, were released into the environment. What followed wasn't theoretical. It was a preview of what climate change is unleashing across the Arctic. 2,649 reindeer died. 36 people fell ill, among them a 12-year-old boy. He didn't survive. This was the first confirmed death from a pathogen released by thawing permafrost in the modern era. Not the last, just the first we documented. The outbreak forced authorities to quarantine the region, destroy thousands of reindeer carcasses, and vaccinate entire communities. But anthrax is just one pathogen we know about, one we have treatments for, one we understand. What happens when the thawed pathogen is something we've never encountered? Something for which no vaccine exists? Something our medical infrastructure isn't designed to handle? The Siberian outbreak proved the mechanism works. Frozen ground thaws. Ancient pathogens emerge. People get sick. People die. The question isn't whether this will happen again. It's what else is waiting beneath the ice. Number 5. Smallpox and Spanish flu. The genetic signatures of history's deadliest pandemics preserved in frozen graves. In 1997, researchers exhumed the body of a woman from permafrost on Alaska's Seward Peninsula. She'd been buried in a village cemetery. Her lungs contained genomic material from the influenza strain responsible for the 1918 pandemic, the Spanish flu, the virus that killed an estimated 50 million people worldwide. 2012, Siberia. Scientists confirmed that 300-year-old mummified remains contained genetic signatures of smallpox, the virus that killed hundreds of millions throughout human history before being eradicated in 1980. These weren't complete infectious viruses, just broken genetic fragments. But their presence raises a disturbing question. If partial sequences survived this long, could complete viruses exist deeper in the permafrost, in colder regions, in bodies buried under different conditions? Smallpox survives well when frozen at negative 20 degrees Celsius. Not for centuries, according to current understanding, but we keep finding exceptions to what we thought was possible. The expeditions that recovered these samples weren't looking for bioweapons. They were studying historical pandemics, trying to understand how these diseases evolved, how they killed so efficiently. But each excavation in permafrost regions carries risk. Disturbing frozen ground means potentially releasing whatever was buried there. And we're disturbing more frozen ground every year. Number 4. The 13 zombie viruses, proof that permafrost diversity exceeds every prediction. In 2020, investigators in Siberia published preliminary characterizations of 13 new viruses. They'd been isolated from 7 different ancient permafrost samples, one from the Lena River, one from Kamchatka, others from sites scattered across the Russian Far East. These viruses represented 5 different clades: Pandoravirus, Cedrit virus, Megavirus, Pacman virus, plus a new Pithovirus strain, each family with distinct characteristics, different replication strategies, different structures, different genetic compositions. 3 belong to a recently proposed Cedrit virus clade, a new genus within the Pithoviridae family. 1 came from the Lena River sample, another from surface permafrost in Kamchatka, a third from mud flowing into the Kolyma River at Duvany Yar, where thawing permafrost layers of mixed ages were collapsing into the water. The diversity was staggering. Before these discoveries, scientists had published only 2 major studies on live viruses from permafrost: Pythovirus in 2014, Mollyvirus in 2015. The scarcity of reports created a false impression that zombie viruses were rare, exceptional finds, not a widespread threat. The 2020 study shattered that illusion. These viruses weren't rare. They were everywhere researchers looked. Every sample contained multiple viral families. The permafrost wasn't hiding occasional survivors. It was preserving an entire prehistoric viral ecosystem. And we're releasing it. 4 sextillion microbes per year, according to estimates published in Environmental Sustainability. That's a 4 followed by 21 zeros, released annually as permafrost thaws. Most are bacteria and archaea. But viruses are part of that count. Ancient viruses with no modern counterparts, no known hosts, no place in current ecosystems, being released into a world that's completely different from the one they inhabited when they were first frozen. Number 3. Tibetan glacier viruses, evidence that the threat extends far beyond the Arctic. A research team drilled an ice core from the Gulia Glacier high on the Tibetan Plateau. 310 meters of ice, layers dating back 41,000 years. They divided it into 9 sections, extracted DNA from each one, and sequenced every organism they found. 1,700 virus species, exclusively infecting bacteria and archaea, waxing and waning over thousands of years as climate shifted between cold and warm periods. The viruses differed markedly between eras. A distinct community formed during the most dramatic climatic shift, right at the end of the last ice age 11,500 years ago, when temperatures spiked, when ice sheets collapsed, when entire ecosystems transformed within centuries. This discovery proved something crucial. Ancient viruses aren't confined to Arctic permafrost. They're preserved in glaciers worldwide, the Himalayas, the Andes, the Alps, anywhere ice has persisted for millennia, and those glaciers are melting, not slowly, catastrophically. Mountain glaciers that took thousands of years to form are disappearing within decades, each one releasing whatever viral cargo it held, into watersheds, into soil, into ecosystems with no evolutionary preparation for what's coming. Understanding how ancient viruses dealt with past climate changes might help predict how modern viruses will respond to current warming. But it also reveals how many unknowns we're releasing, how many viral families we've never studied, how many potential hosts we haven't identified. The Tibetan viruses infected bacteria and archaea, not humans, but that's exactly what researchers said about the Siberian giant viruses before they started finding diversity in replication strategies that mirrored human pathogens. Every new discovery reveals how little we actually know about what's preserved in ice. Number 2, deep-sea sediment viruses, the threat nobody saw coming. December 2024. A research team published findings that should have made international headlines. It didn't. The study examined ancient viruses trapped in deep-sea sediments. Not permafrost, not glaciers. The ocean floor. Sediments dating back 1,900 to 17,300 years. They extracted viruses from 106 different samples spanning that entire range. Then they did something nobody had thought to test before. They exposed those ancient marine viruses to modern terrestrial soil bacteria. The results were catastrophic. Viruses purified from 9 of those sediment samples could infect soil bacteria. Not just infect them. Destroy entire bacterial communities. Disrupt soil functions completely. These were marine viruses from the ocean floor, successfully attacking terrestrial ecosystems they'd never encountered before. Cross-environment compatibility that shouldn't exist. The implications are staggering. We've spent years worrying about Arctic permafrost releasing ancient pathogens into northern ecosystems. Meanwhile, deep ocean mining and drilling operations are disturbing sediments that have remained undisturbed since before recorded history began, bringing up material from depths where pressure and darkness have preserved viral communities we've never studied, releasing them into surface waters, into coastal zones, into environments with zero evolutionary preparation for what's coming out of the abyss. The biosecurity risk of benthic viruses had never been evaluated on a global scale before this study. We simply didn't consider it a threat. The ocean seemed too vast, too diluted, too different from terrestrial environments to pose cross-contamination risks. We were catastrophically wrong. Here's what makes this discovery particularly terrifying. Human activity in deep oceans is accelerating. Mining operations targeting rare earth minerals, drilling for oil in deeper waters than ever before, scientific expeditions disturbing sediment cores. Every operation brings ancient material to the surface. We have no protocols for this, no containment procedures, no risk assessment frameworks, because until December 2024, we didn't know we needed them. Number 1. The immune system problem. Why ancient viruses might be unstoppable. Scientists worry about zombie viruses for reasons that go far beyond their age or preservation. The real threat is recognition. Our immune systems operate on memory. Your adaptive immune response relies on previous exposure to similar pathogens. Antibodies. T cells? The entire framework of acquired immunity depends on your body having encountered something comparable before. If a virus from the Ice Age reappears, your body might not recognize it at all. Not as a variant of something known, not as a distant relative of a modern pathogen, as something completely foreign. Your adaptive immune system would be starting from zero, but surely your innate immune defenses would respond, right? The first-line mechanisms that identify general patterns associated with microbes. The problem is, those patterns evolved based on modern pathogens. Ancient viruses might display molecular signatures our innate immunity doesn't recognize as threatening. Researchers simulated thousands of scenarios of ancient pathogens being released into modern ecosystems. The modeling accounted for variables we can measure: transmission rates, environmental stability, host availability. In the worst-case scenarios, up to 1/3 of host species were destroyed. Not infected, destroyed. Entire populations eliminated because nothing in their immune repertoire could mount an effective response fast enough. The situation becomes exponentially worse if the revived pathogen affects plants or animals for which we have no specific treatments ready, no vaccines in development, no therapeutic protocols, no understanding of pathogenesis. An ancient unknown virus causing agricultural collapse or livestock die-offs would face zero medical countermeasures. We'd be watching it spread with 19th century tools. The Arctic is warming 4 times faster than the global average. That's not a projection, that's current measurement. Permafrost that's remained frozen for millennia is thawing within single human lifetimes. And here's the accelerating feedback loop nobody talks about enough. Permafrost contains 1,500 gigatons of carbon, twice what's currently in the atmosphere. As it thaws, that carbon releases as carbon dioxide and methane. Those greenhouse gases warm the planet further, which melts more permafrost, which releases more carbon, which causes more warming. We're not just opening a biological time capsule. We're creating conditions that ensure it keeps opening faster every year. Meanwhile, global warming is making Arctic regions dramatically more accessible. New shipping routes through previously frozen waters, resource extraction in areas that were too remote to exploit economically, mining operations, oil drilling, construction projects, more people working in regions where ancient pathogens are actively being released. The public health risk isn't theoretical anymore. In 2016, an anthrax outbreak in Siberia's Yamal Peninsula killed 2,649 reindeer and hospitalized 36 people. A 12-year-old boy died. The outbreak was traced to a reindeer burial ground where animals had died from anthrax over 70 years earlier. An unusually hot summer thawed the permafrost, released spores from frozen carcasses. Within weeks, thousands of animals were infected. That was one pathogen, one region, one summer. We've now confirmed that permafrost contains thousands of viral families, multiple replication strategies, diverse host ranges, preserved across the entire Arctic, all of it thawing simultaneously. The 48,500-year-old Pandora virus Yedoma proves these organisms remain viable across timescales that dwarf human civilization. The Tibetan glacier viruses prove the threat extends far beyond polar regions. The deep-sea sediment viruses prove we haven't even identified all the environments where ancient pathogens are preserved. We're conducting a planetary-scale experiment in microbial resurrection, releasing organisms that haven't interacted with modern ecosystems since the Pleistocene. And we have no idea what most of them are, what they infect, or what happens when they encounter a world that's completely different from the one they left. The zombie viruses are already awake. We're just beginning to understand what we've unleashed. The terrifying reality is that we're not discovering these viruses because we've gotten better at looking. We're discovering them because they're being released in quantities impossible to ignore. Every expedition into thawing permafrost finds new viral families. Every ice core reveals thousands of species we've never cataloged. Every deep-sea sediment sample contains pathogens that can cross environments we thought were separate. This isn't a problem we can solve by stopping research or avoiding disturbed ground. The release mechanism is already running. 4 sextillion microbes annually from permafrost alone. Glaciers collapsing worldwide. Ocean sediments disturbed by expanding industrial operations. The biological time capsules are opening whether we're ready or not. What makes this moment in history uniquely dangerous is the convergence. Climate change accelerating viral release. Global travel ensuring rapid pathogen spread. Dense human populations providing abundant hosts. Industrial expansion into previously untouched regions. We've created optimal conditions for ancient diseases to find modern victims. The scientists studying zombie viruses aren't trying to scare you. They're trying to warn you. Because once these pathogens establish themselves in modern ecosystems, once they find susceptible hosts and begin circulating, we can't put them back in the ice. The Pleistocene is reaching forward across 50,000 years, and we're about to discover whether our species can survive what we've awakened.